This is part 3 of chapter 5, where we are continuing our tour of eukaryotic cells, discussing different types of organelles. And here we have the mitochondria, which is the site of cellular respiration. And cellular respiration is the process whereby the energy that is stored in carbohydrates and sugars like glucose molecules inside of cells is converted into molecules of ATP, which is really the preferred energy molecule that can be used very easily to power all the work of a cell. Um, and we have a whole chapter coming up about cellular respiration, so I don't want to go too much into it at this point. We will come back to it, but I do want you to be aware of the structure of mitochondria because that's going to come back into play when we talk about cellular respiration. And if you look at the cartoon of the mitochondria here, you can see it has two membranes. So it has a smooth outer membrane. There's then a space and then an inner membrane. And that inner membrane is very highly folded. So you can see if we look at it in cross section here, all these foldings. Um, and then the very interior, the yellow portion here, would be considered the matrix of the mitochondria. These folds, as you can see labeled here, are called cristae. And really the whole point of these folds is to give a very high surface area because as we'll see when we talk about cellular respiration, a lot of the machinery that's actually producing ATP is embedded within this inner membrane. So the more of that inner membrane that's present, basically the more ATP could be produced. So it's all about increased efficiency. Um, let's shift over to a plant-specific organelle. I mentioned plastids earlier in the chapter, and really the main plastid we're going to focus on are chloroplasts, which hopefully we're all at least somewhat familiar with. So obviously chloroplasts are where photosynthesis happens. They contain that lipid chlorophyll, right, that photosynthetic pigment. Um, the structure of a chloroplast does have some similarities to mitochondria. So we have a double membrane again. In this case, both membranes are smooth. Um, there's a fluid inside the mitochondria called the stroma. And then we really have what we should consider to be really a third inner membrane in this case, which is called the thylakoid membrane. And it's got this unusual structure where it it looks like it's almost made up of like individual little coins or poker chips stacked up. So you'll see each one of these little discs called a thylakoid, but really all this green stuff you see here is all thylakoid membrane. Um, but each of these discs are interconnected, so this is all continuous membrane. You can see there's little strands connecting the various stacks. Each of these stacks is called a granum. Um, and this looks green because this is where all the chlorophyll is located. So chlorophyll is capturing the energy in sunlight, and then in chloroplasts we're able to use that energy to generate sugars. And again, we have a chapter coming up all about photosynthesis, so we will revisit the structure of chloroplasts here and talk about how that structure ties in with the function in the near future. So I mentioned, we're primarily just going to focus on chloroplasts in this class, but I did want you to be aware that there are some other types of plastids found in plants as well. So for example, there are what are called chromoplasts. These are plastids that basically contain other types of pigment molecules. Chromoplasts are what will give flowers their unique colors. There are leucoplasts, which are plastids that store starches and fats. You can see a leucoplast here with these big starch grains show up as these white blobs on the electron microscope inside of that structure. Uh, so just be aware there are other types of plastids. And while we're talking about plants, let's talk about another plant-specific organelle, which is the vacuole. And vacuoles take up a lot of real estate inside plant cells. So in fact, in some plant cells, like 98% of the interior of the cell is vacuole, and all the cytoplasm is basically just squished around the side. Here, I'll jump ahead, show you a picture here. You can see this is a picture taken with an electron microscope of this plant cell. You can see some chloroplasts here, the big dark structures, but this big white area in the middle of the cell is all vacuole. So what is the vacuole doing? It's taking up a lot of space. It could be used for storage, storing waste. Sometimes even toxic compounds will get stored in the vacuole. Those might be used, for example, to try to deter herbivores from eating the particular plant. They're also very important just in terms of providing some structure to plant cells. So typically that vacuole is filled with fluid, right, primarily water, and the outward pressure of the vacuole pushing out on the plant cell creates this phenomenon called turgor pressure. So this is what would, for example, prevent a soft stem plant from being wilted. If you have a plant that is wilted, that is because the vacuoles are not full of fluid in that plant, and the cells are a little bit soft and a little too flexible, so the plant flops over. You fill the vacuoles back up with water, you get this pressure that will allow it to stand up. We were just talking about how pigments could be stored in plastids. They can also be stored in vacuoles in some cases. And there are examples where there are actually some, very biologically active molecules like enzymes that might be stored in vacuoles in certain instances as well. All right, so I want to switch gears a little bit here and talk about a part of the cell that's not really truly an organelle. I don't think it necessarily gets talked about in a lot of biology classes, but it is a critically important part of the cell. And my research background, my PhD, all dealt with this topic, so it's something that I definitely don't want to skip over. But I'm referring to what's called the cytoskeleton here. And so if you think of your own skeleton, right, an animal skeleton, the job of that skeleton is to provide structure and support to your body, right? You've got muscles attached to your bones, and it's giving you your distinct body shape. And the cytoskeleton does play that role inside of cells as well. So you can think of this as, miniature skeleton inside the cell. It does help maintain cell shape, does provide some support, but it has lots of other jobs as well. So it could be used to anchor things in place, can also be used as almost like railroad tracks to move things around within a cell. And in fact, the movement of organelles and vesicles along some of these cytoskeletal tracts will result in this phenomenon called cytoplasmic streaming, which is sort of just like it sounds. It's the cytoplasm sort of being stirred up and streaming around inside of a cell, which can help aid in diffusion of molecules within a cell. And as I'll mention, the cytoskeleton can also interact with extracellular structures, so outside of the cell, can be used for anchoring cells or cell-to-cell type of interactions. And unlike an animal skeleton, the cytoskeleton is actually very dynamic, so it can change very quickly. It could shrink or grow, branch, so it's constantly in flux. And there's three basic types of cytoskeleton I want you to know about. There's what are called microfilaments, then intermediate filaments, and microtubules. And you can see each of those illustrated in cartoon format down here. You've got microfilaments on the left, intermediate filaments in the middle, microtubules on the right. And below those cartoons, you can see actual pictures taken with fluorescent light microscopes where they have the microfilaments, intermediate filaments, and microtubules labeled with fluorescent protein inside a single cell. And you can see there's a lot of these structures in one cell, right? This is just the microfilaments in red, just the intermediate filaments in green. And imagine, cells are going to have all three of these inside them at the same time. So, when I show you these cartoons of cells, the cytoplasm kind of looks like just an empty space, but in reality, it's really going to be packed full of these cytoskeletal fibers and structures. So I'm going to go through the three of these briefly and just talk about, um, you know, what is the main job of these different types of cytoskeletal components and what are they made of. So let's start with microfilaments. And there's a few important jobs of microfilaments. They can be used for anchoring things in place, can be used in actually moving cells around or moving things around within a cell. So very important for movement, important for determining cell shape. And if you've ever studied muscle contraction, your muscle fibers are basically made up of alternating rows of essentially microfilaments and then this motor protein that's called myosin. And what will happen is these motor proteins will all change their shape simultaneously and allow these microfilament fibers to kind of slide past each other, and you'd get this shortening of the muscle fiber or contraction, and then the myosin motor proteins would all relax and the filaments would slide back past each other in the opposite direction, and your muscle would likewise relax. And so there are other motor proteins aside from myosin that can be used to actually move things around on these tracks, on these microfilament tracks. So you can think of the motor proteins almost as little trains or little locomotives that would be moving things around within the cell. Excuse me. Microfilaments are made of a protein called actin. So microfilaments are a polymer. You see actin referred to here as the monomer. So these little circular proteins, and we just link a bunch of these together in a chain, and that is the microfilament in a nutshell. And we can grow this filament longer by adding more actin monomers, or we can shorten it by removing them. So here's an example of how microfilaments would be used to power the movement of a cell. And this would be important in embryonic development in animals. You have cells moving all over the place. This is an important part of disease, so cancer cells, right? One of the reasons cancer is so scary is because cells can leave a tumor and migrate away into the bloodstream and then establish metastases throughout the body. So this phenomenon would be powering that element of cancer as well. But what you see here is a cell, and you'll notice just under the plasma membrane you have this network of microfilaments. And those microfilaments are associated with this myosin motor protein. This is almost like a miniature muscle tissue, almost essentially, just under the surface of the cell here. And what will happen if this cell wants to move, and in this case it's going to move off to the right, you will get the microfilaments that are at the right side of the cell, just under the plasma membrane, will all start elongating. So we will add more of those actin monomers. The microfilaments will grow longer and actually push the plasma membrane forward, forming what's called a pseudopod or a false foot. And then while that's happening at the back end of the cell here at the left side, you will get basically a contraction of that actin and myosin structure. So the myosin motor proteins would all change their shape simultaneously, just like I talked about would happen in a muscle fiber, and will basically be squeezing the back end of the cell here due to that contraction, kind of forcing everything off to the right. So the combination of those two actions is going to cause this cell to move to the right in this case. I mentioned that microfilaments were important for cell shape. Here you have microvilli cells that have a very unique shape. So these are cells that are found lining the small intestine in your gut, and they're have these long finger-like projections you can see here. And so, the microfilaments inside these microvilli cells are basically helping allow them to stand up, so the microvilli don't just flop over. And you can see the actin filaments in purple here, basically just running parallel to the length of the microvilli. And then you have other little proteins in here, cross-linking proteins they're called, that are essentially just serving as scaffolding to help maintain that structure. And the purpose of these microvilli cells is just to provide a large surface area for absorption of nutrients as digested material is passing through your gut. So microfilaments important for movement of cells. We just saw an example of that. Important for maintaining the shape of cells. What about intermediate filaments? Well, intermediate filaments seem to have arguably less roles than either microfilaments or microtubules. They primarily seem to be important for anchoring things in place inside of cells and for helping to resist tension on cells. So intermediate filaments are made of a protein called keratin, and keratin is the same protein you find in your fingernails and in your hair, right? So it's this very tough, fibrous protein. These keratin fibers get kind of wound around each other. You can see up here in the cartoon, and then these structures get assembled into the filament itself. And so because intermediate filaments are made of this tough fibrous keratin, really you could kind of think of these structures, these filaments, as almost like the springs inside of a mattress, right? So they're resisting that tension. So if a cell is pushed on or bumped into, it's not going to just flatten or explode. So we know the least about intermediate filaments. So not a whole lot to say there. We do know a lot about microtubules. Microtubules are made of a protein called tubulin. That kind of makes sense. There's actually two flavors of tubulin, alpha tubulin and beta tubulin. So these two protein monomers get brought together to form what's called a dimer, di meaning two. And then those dimers get assembled into the microtubule, which is literally a hollow tube. I'll show you a picture in a second where you can see this in cross section, but you could actually look straight through the microtubule. So what do microtubules do? Well, they can also act as tracks for motor proteins to move things around inside of cells. I'm going to show you an example where they're very important for powering cilia and flagella, so they can also help drive cell movement if a cell happens to have one or both of those types of structures. And just as I talked about with microfilaments, these microtubules can grow or shrink just by adding or removing dimers. With the intermediate filaments, let me go back one slide here, these are not believed to change as much as microfilaments or microtubules, because these larger subunits don't seem to be able to be added or removed as easily. All right, so here's an example of microtubules inside of cilia and flagella. So again, this would be used to power either cell movement or movement of things past a cell. Typically, flagella will be used to propel cells, whereas cilia are typically used to move things past stationary cells. So, for example, your upper respiratory tract is lined with cilia that are constantly sweeping dust and debris out, right? So your lungs don't fill up with dirt and dust. Also helping to keep bacteria and viruses out. But regardless, if you take a cilia or flagella and cut it in cross section like this, and then look at that cut end, you will see something like this, um, which is this very distinct and unique arrangement of microtubules. So each one of these black rings that you see here is the cut end of a microtubule. So now uh, you're looking down through the hole in the middle of the microtubule here. And you get this arrangement, what's called the 9+2 array of microtubules. So you get 9 pairs or doublets around the outside, and then a central pair in the middle. So 9 pairs and then 2 in the middle here. And again, you find that in basically every cilia and flagella. So, why that unique arrangement? Well, what's happening is those microtubules are all connected to each other with this motor protein called dynein. And these dynein molecules will all try to change their shape. Well, they won't try, they will change their shape kind of simultaneously. And when they do that, that would try to slide the microtubules basically past each other. So the one on the right here would want to slide down. But it's unable to do so because there's also these cross-linking proteins between the microtubules. So instead of sliding past each other, what will happen, as you see on the right here, is the whole structure will just bend instead. So the dyneins are basically driving the bending of these microtubules. And so that bending is what's going to power the waving of the cilia or flagella. I mentioned that microtubules, very much like microfilaments, could serve as these tracks for the movement of motor proteins. And here's a little cartoon illustrating what that would look like. So here's your microtubule in green. You have the motor protein in purple here. And the motor protein can actually carry some cargo. So at one end it can bind to the microtubule. At the other end it will bind to something like an organelle or a vesicle. And using energy from ATP, that motor protein will change its shape and can literally walk along the microtubule. Or as I said, there's something similar happening along microfilaments as well, and thereby carrying that cargo along. So if you think back to earlier in the chapter, like where we were talking about the endomembrane system, and I was discussing the fact that these vesicles could move from the ER to the Golgi, and then from the Golgi to the plasma membrane. In those cartoons that I showed you, it looks like the vesicles are just kind of floating around in the cytoplasm. But in reality, they are probably being carried along either microtubule or microfilament tracks by these motor proteins. Okay, so more than you ever wanted to know about the cytoskeleton, but just be aware of basic structure and function of those 3 different types. All right. We talked about a couple of plant-specific organelles. I wanted to revisit centrioles, which were an animal-specific organelle. And you might remember I mentioned earlier that centrioles are important for growing the spindle that forms during mitosis. And it turns out that spindle is made out of microtubules. So essentially you could think of centrioles as sort of like seeds for growing those microtubules that are going to be important for powering mitosis. Okay, so I think we have exhausted all the organelles that you're going to find inside cells. So at the end of the chapter here, I want to talk just for a minute about what's going on outside of the cell, so outside the plasma membrane. And of course, in plant cells, you have the cell wall, which is this thick structure made of cellulose fibers. Also contains a lot of other sugars and proteins. And you probably don't spend a lot of time thinking about plant cell walls, but if you do, it's tempting probably to think of it as just kind of this mess of cellulose fibers. And, I suppose in some sense that's true, but there actually is some organization to the cell wall as well, which is what I wanted to point out here. So you can see in this picture taken with an electron microscope here, you've got two plant cells in the light gray, the cytoplasm of the cells, and the cell wall in the darker gray here. And you can see there's like different layers, right? Pretty organized layers to the cell wall, so you don't have to memorize the names of the parts here. But just be aware there is some organization to it. And there are also some pores that will basically go all the way through the cell wall, um, connecting the cytoplasm of two plant cells. And these pores or channels are called plasmodesmata. So this would allow small molecules to actually move from one plant cell to the next through those plasmodesmata. Now, what about animal cells? Animal cells don't have cell walls, but they do have what's called the extracellular matrix, which I think it's fair to think of as sort of the light version of a cell wall. So the extracellular matrix is also very fibrous, so it's got proteins like collagen, um, things like proteoglycans that are sort of dense and fibrous, just not quite as dense as the cellulose fibers you find in a plant cell wall. But you can actually see some pictures here of what that might look like. And so every animal cell, every cell in your body is surrounded by extracellular matrix. And the jobs of the extracellular matrix are pretty varied, but they're helping to hold cells together into tissues. You find lots of extracellular matrix in things like bone and cartilage and skin. The extracellular matrix can act to filter things out that may be moving between cells. If you have a cell that's going to move, so like we were just talking about a few slides back, that cell is moving basically on and through the extracellular matrix. So it's having to interact with that matrix as it goes. And any signals that might be arriving at cells would be passing through that extracellular matrix. So, even though it's definitely not the most glamorous part of this chapter, there are a lot of important roles that the extracellular matrix is involved in. All right, so last point is just a little bit of speculation in terms of how did we get these incredibly complex cells with all these organelles, because all the evidence suggests that life on Earth started as prokaryotic cells, right, which lack all these membrane-bound organelles. So how do we go from these arguably simpler prokaryotic cells to these more complex eukaryotic cells? And again, we don't really know because nobody was around to observe this happening. But it's not too hard to imagine, for example, the beginnings of formation of nucleus or ER simply by folding in the plasma membrane. So imagine we've got our primitive cell up here at the top, right? No organelles. We've got our DNA just floating around in the cytoplasm. So imagine you start folding in the plasma membrane into the interior of the cell. If we do that enough times, all of a sudden we've kind of pinned the DNA into the center of the cell, right? We've got it surrounded by a membrane in what could be considered basically a primitive nucleus. And as we talked about in this chapter, the nucleus is continuous with the ER, right? The ER is simply just folded membrane. So, these membranous folds look very similar to ER and even to Golgi for that matter, right? So it's speculation, but it doesn't seem, too far into left field to think that this is how we could have ended up with nucleus and ER and probably Golgi. One thing that is pretty clear, there's a lot of evidence for, is the fact that two of the organelles we talked about, mitochondria and chloroplasts, were almost certainly at one point free-living bacteria that develop this symbiotic relationship inside of another cell. And so this is called the endosymbiosis theory, endo for inside. And the basic idea here is that these free-living bacteria, one that was essentially really specialized for ATP production, or one that was capable of photosynthesis, found themselves being taken up inside of another cell, and instead of being digested, were able to survive inside that cell, and there was presumably benefit to both parties here. So in the case of the photosynthetic cyanobacterium, that bacteria is getting nutrients from the host cell, and the host cell is maybe getting some sugar that's being produced by photosynthesis from that cyanobacteria, so both benefit. And then at some point, what is believed to have happened is some of the DNA, some of the genetic material from that cyanobacteria was actually transferred into the nucleus of the cell. And so at that point, that bacteria is basically trapped. It doesn't have all of its own DNA anymore, so is incapable of leaving. And it's really dependent on that host cell to provide it some of the proteins that it needs to function. So, okay, so that may sound crazy. What is the evidence for that? And there's really two things. One is if you look at mitochondria and chloroplasts, they look a lot like bacteria just in terms of their shape. So that's fine. But really the strongest evidence is those two organelles actually have their own DNA, which none of the other organelles have, right? And if you look at that DNA, if you sequence it, it looks way more like bacterial DNA than eukaryotic DNA. For one thing, it's circular DNA. Bacteria have a circular chromosome. We see genes there that we clearly see in bacteria and not in eukaryotes. So that endosymbiosis theory is supported by a lot of evidence. And I remember learning that as an undergrad, and I think my head almost exploded. I was like, what? That is craziness. All right, thanks for hanging with me. I know Chapter 5 was a long one. In Chapter 6, next time we're going to focus on cell membranes. We'll dive a little bit deeper into the structure and function of cell membranes and talk about how things cross membranes, get in and out of cells.